Hi students, myself Dr. Ganesham Chavan. Today we are having discussion on the topic the muscular system. Students, all over the body we are having various types of muscles which are having various functions in our body. They are major function to do the locomotion, to do protection and to transfer the impulses from one part to the another part. So in all over the body, if you see various muscles are there. For example, the frontalis muscles, the masseter muscle, deltoid muscle, bicep, tricep muscles, brachialis muscles. Like this, in all of the part, muscles are available. Without muscle, we cannot move, we cannot walk, we cannot run. There are four characteristics which are associated with the muscle tissue. In that first is excitability. So whatever the muscles are there, so the muscles can be excited with the help of neuron. Means the action potential is going to be generated and which will convert the electrical energy into the chemical energy in the form of neurotransmitters. And they are firing these neurons, means the impulse generation will occurs and the impulse transmission occurs from one neuron to the another neuron, and which are give rise to the excitability of your muscle. So tissue can receive and respond to the stimulation, and stimulation occurs in the form of the neurotransmitters release. Next is contractibility, contraction of muscles. So this contraction of muscles occurs due to the action potential. So whatever the neurons which has been stimulated, so their stimulation and impulse generation will lead into the contraction of the muscles. So muscles will contract regularly after the interval of the time, and the tissue can shorten and thicken. Means the muscle tissue will shorten, means the contraction occurs, and thicken, and if they will relax, so they will straight. So in this fashion, in the heart, the continued contraction and relaxation occurs, and also in your skeletal muscle the contractility can be checked. The extensibility, so in this the tissue can be lengthened, means the if you are giving stress on the muscles, so the muscles can be stretched, means their size can be lengthened, increase. So that typical property is called as extensibility. So next property is elasticity, means after the complete extension, after the complete elongation, when the muscle return back to its normal position, so that typical condition is called as elasticity. Means after contraction or lengthening, tissue always want to return to its resting state. The characteristics of muscle tissue enable it to perform some important functions. The muscles are having some important functions that we will discuss one by one. The first important function is to do movement, so body voluntarily or involuntarily. Now in case of skeletal muscle, so our body is involved into the various type of movements. When you are moving our body with the help of legs, or when you are doing some actions with your hand or face, when you are running, walking, when you are jogging, so in that movement our muscles are involved majorly. And in case of involuntary movement, the movement of our gut, the movement of our urinary bladder, when they are doing movements, after that the gut movement, the peristaltic movement which occurs. What is peristaltic movement? The movement which occurs into the small and large intestine when the repetitive episodes of contraction and relaxation occurs, and they are responsible for movement of food from our mouth to the anus. So that peristaltic movement is responsible for that. So next is maintaining posture. So whatever posture we are holding, either it is standing posture or sitting posture, or it is in tilted posture. So the posture which we are holding, that only occurs due to the contraction of your muscles. If your muscle won't contract, we can't able to stand or sit or do any function. What will happen if we are not able to maintain the posture? We will simply lay down on the bed. We cannot sit even. Next function is supporting soft tissues within the body cavities. So the muscles are revolving or wrapped over the soft tissue, and due to which they are protecting our internal bodies. Now for example, the muscle which are the sphincter muscle which occurs at the stomach, there are two muscles. One is the cardiac muscle and another is a pyloric muscle. So these sphincter muscles are responsible for supporting the soft tissues in the body. So the hollow part of our stomach is going to be supported by our muscle, the cardiac sphincter and the pyloric sphincter, like that. So they are supporting the soft tissues. Now in case of our pectoral girdle, the pectoral muscles are supporting our soft tissues of bone. So they are also supporting our pneumatic bones and as well as our lungs as well as the diaphragm. Another type of muscle, for example, rectus abdominis muscle of our abdomen, so they are supporting our peritoneum, they are supporting our small intestine. Next is guarding entrances and exit of the body. So for example, the pyloric sphincter of our stomach, they are guarding the entrances, means they are not allowing. Another example is that urinary bladder. So our urinary bladder is having the muscles which are protecting to close and to open that typical opening of our urinary bladder from the urethra, which are protecting their entrance, means they are opening voluntarily. When we want, we can do urination. When we don't want, we can suppress them, we can control them. Maintaining body temperature. So our muscles are able to produce heat inside our body. So they are doing the process of thermoregulation in our body. In this fashion, they are maintaining our body temperature. Types of muscles. We are having three types of muscles. One is skeletal muscle, the muscle which are available to our skeleton. They will be axial as well as the appendicular skeleton. They are attached to our bones. Next is cardiac muscle, the muscle which are available in our cardiac, in our heart. The skeletal muscles are striated muscles. The cardiac muscles are also striated muscles, whereas the smooth muscles which are available in our respiratory system, in our uterus, in our gut, means intestines, our mucosa. So that muscles are non-striated muscles. Now here, first skeletal muscle tissue. They are associated with the attached to the skeleton and under one conscious control. The microscopically the tissue appears striated. So here we are getting the striations are available. The cells are long, cylindrical and multinucleate. So here you can see how the muscles are there and how they are looking. So here you can see the striations are here. So these are called striations. Next is cardiac muscles. They make up the myocardium of the heart. Means in the heart we are having the myocardium muscles, and these cardiac muscles are responsible for pumping of our heart. So whatever the contractility is there, that only occurs due to the myocardial muscle. So unconsciously they are controlling, means they are not under control of our will. So they are pumping the heart, means they are increasing and decreasing force of contraction of the heart, and they are maintaining our heart rate at the 72 beats per minute, and also they are maintaining our blood pressure at 120 by 80 mmHg. Cells are short, branching and have a single nucleus. The cells connect to each other at intercalated disc. So these are also having the striations. Here you can see these are also having the striations. They are also having the nucleus and also having the intercalated disc. So through this the muscles are connected. Smooth muscles, they make up the wall of organs and blood vessels. The tissue is non-striated and involuntary. The muscles are not having striations. Cells are short, spindle shaped, and have a single nucleus tissue. Tissue is extremely extensible while still remaining ability to contract. So these muscles are contacting according to the involuntary system, autonomous nervous system. So they are not under control of our will. Now we will discuss the anatomy of skeletal muscle. So here that skeletal muscle we can see. Now if you will see the anatomy of skeletal muscle, in that the muscles which are available in our arms, that you are see here. So this is the muscles of your arm. So in that muscles the tendons are there. What is tendon? Tendons are nothing but the elements which attach your muscle to your bones. So tendons are having great role into the muscles for holding the muscles, to attach the muscles to the bone. Now this is the part or is the covering of the muscle. It is called as epimysium. It is the outermost layer of the dense irregular connective tissue, and it is encircling the entire muscle. Now after this outermost material, now here you can see they are just below that epimysium. So they are called as perimysium. So what is perimysium? So it is also a layer of dense irregular connective tissue, but it surrounds group of 10 to 100 or more muscle fibers, separating them into bundles called fascicles. Many fascicles are large enough to be seen with the naked eyes, so they give a cut meat its characteristic grain. Now after this perimysium, now we will see this perimysium is surrounded by these fascicles. Now after this is the endomysium, which is the internal part of muscle. So it penetrates the interior of the each fascicle and separate individual muscle fibers from one another. The endomysium is mostly reticular fibers. So you can see this is the reticular fibers, and the skeletal muscle fibers are cells are there, and it is surrounded by the endomysium. Now here in this microscopy of this muscle fiber, here you can see these are the skeletal muscle fibers are there, and this is the nucleus, and these are the striations. The striations you will get in case of the skeletal muscle as well as in the cardiac muscles. Now in case of the microanatomy of the muscle fibers, so here you can see sarcolemma. These sarcolemma are nothing but the cell internal element which is responsible for release of calcium. So they are the multiple nuclei of skeletal muscle fibers which are located just beneath. Now transverse tubules. So these transverse tubules are nothing but the thousands of tiny investigations of the sarcolemma. This sarcolemma. Sarcoplasmic reticulum. This sarcoplasmic reticulum are responsible for release of calcium from your muscle, and this sarcoplasmic reticulum are fluid filled system of membranous sacs, and they encircle each microfibril. The terminal cisternae. These terminal cisternae are dilated and sacs of the sarcoplasmic reticulum, but they are against the T tubule. This is the triad. Now after this triad, this is the nuclei, which you can see very distinctly. This is the mitochondria, which gives energy to the cell of our muscles. This is the myoglobin. The myoglobin, it is nothing but the red colored protein of our muscle. So it is called as myoglobin. Almost 5% of the total protein it is available into our muscles. It is a microfibril. This one. This is the thick filament and this is the thin filament. If you see this microfibril, these are the structures and they are at high magnification. The cytoplasm appears stuffed with the little threads. So they are thread-like structures. That's why they are called as microfibrils. Fibrils means fibers, means thread. Microfibrils. These microfibrils are about 2 micrometer in diameter and extended the entire length of the muscle fibers. Now here in the thin microfilament, now here you can see how these muscles are bended and extended, and they are having these spring-like structures also for relaxation and extensions. For the they are giving the complete area for relaxation and extensions. This is the Z line, Z disc is there. Now here in this is the tropomyosin, so which are temporarily attached to the actin. This is the active sites, and this actin is the active sites, and it is temporarily attached by this tropomyosin, which can be released afterwards during the process of contraction. These are the actin molecules. These actin molecules are responsible for process of contraction, so they will bind to the myosin. Now here this is the thick filament. Here you will find this M line. In early thin filament there was the Z line or Z disc. In this thick filament there is the M line, and this is the myosin tail. This is the myosin molecule, and here it is a zinc shape is there. And in this thick filament we are having this myosin head, and this myosin head is responsible for extension and relaxation. So it has a ATP and actin binding site. So this is the actin binding site which will bind with the actin, and it is also having the ATP, so which will release the ATP. Now here the sarcomere. In sarcomere the various zones are available, various bands are available in this. So in this band, if you will see, this is the thin filament and this is the thick filament. So in that filament, how various zones are available and how they are overlapping, that we can see here. Now this is the Z line, this is the Z zones where there is a process of contraction are involved. So this Z zone is having a great role. This is the A band, this is the Z line. Now if you will see the A band which extend the entire length of the thick filament, whereas the H zone is the center of each A band which contains thick but not thin filament, whereas the M line, this is the M line. So the M line, so name called that it is the middle line of the sarcomere. So the whole sarcomere, if you see, it is a middle line of your sarcomere. And whereas the Z disc is there, so the narrow plate shaped region of dense protein material is called Z disc. which separate one sarcomere from the next. Thus, the sarcomere extend from Z disc to the next Z disc. So, in this session, we have discussed this sliding filament theory. So, the myosin heads attached to actin molecules at the binding site. The myosin pulls on actin and causing thin filament to slide across the thick filaments. Means this thin filament will will slide across these thick filaments toward the center of sarcomere. This is the sarcomere, central sarcomere. So this thin filament will going to be extend over this thick filament. The sarcomere shortens, I band gets smaller, H zone gets smaller, and the zone of overlap increases. In this question here you can see the overlap has been occurred. Means the contraction and filament sliding has been occurred. Means the muscle has been completely contracted. A sarcomere shortens, the microfibril shortens. As the microfibrils shorten, so does muscle fiber. Once a muscle fiber begins to contract, it will contract maximally. This is known as the all or none principle. Either all the muscles will going to be contract or nobody will contract. Means all the muscles will start to contract at the same time. So in this fashion, the all and none principle will be followed here. Now this is the physiology of skeletal muscle contraction. How this skeletal muscle will do its contraction. The skeletal muscle requires stimulation from the nervous system in order to contract. Without nervous system, it is just impossible to do the contraction because these nerves are responsible for the generation of impulses and carry that impulses from the one neuron to the another neuron. So most neurons are cells that causes muscle fibers to contract because they are carrying the impulses. For example, if you are giving order to your leg to throw or to give a firm kick to the football, so that message comes from our brain. So and for accordingly from the brain to the tip of your feet, so that impulses is going to be generated and going to be transferred within a nanoseconds, and you are giving the response very fastly. So in this fashion it will move very fastly, it will run very fastly. Now here you can see it is a neuromuscular junction. So that in neuromuscular junction, how there is role of neurons in that junction. So this is the junction between the nerves and your muscles. So it is a motor end plate of sarcolemma of your muscles, and this is the neuron. It is a this is called neuromuscular junction, and this is the neuron. It is a tilde area of neuron, and this is the area of synapse. This is synaptic cleft, and synaptic end bulb is here. This is the synapse, and this is the synaptic cleft. Where the exchange of neurotransmitter occurs, that place is called as synaptic cleft. And when there is connection between two neurons or the neuron and the acceptor, this is the acceptor plate, that area is called as synapse. Now here the synaptic clefts are available, and these synaptic vesicles are containing the neurotransmitter, for example acetylcholine, dopamine, like that. So they are having these neurotransmitters. So whenever the process of transmission of impulses will occur, in that case the acetylcholine means the neurotransmitter which is into the vesicles, they will release here and they will transfer from this neuron to this muscle end plate, to this motor or plate of sarcolemma. So in this fashion, at the neuromuscular junction, the transmission of neuron will occur. Overview of the event at the neuromuscular junction. Now we will discuss the what are the events that occurs into a neuromuscular junction. First is action potential. What is action potential? Action potential are nothing but they are the process which occurs due to this potential. That's why it is called action potential. Means the action which has carry forward by the influence of potential. It is called action potential. The action potential occurs into our neuron when there is a typical voltage is there, and due to that voltage there is opening and closing of channels, voltage gated channels, the channels which are open and closed, and the channels are responsible for release entry of the various electrolytes. It may be calcium, it may be sodium, it may be potassium, it may be chloride, like that. So an electrical impulse is going to be generated and it's going to be transferred by the process of action potential, and they travel down the axons of the motor neuron to the end bulbs. So means they're transporting that impulses from the axon toward the end bulb of your neuron. The action potential causes the synaptic vesicles to fuse with the end bulb membrane and resulting in the release of acetylcholine, which is a neurotransmitter, into the synaptic cleft. So as we saw here, the acetylcholine will release from this synaptic cleft into your muscles. The acetylcholine diffuses across the synaptic cleft and bind to the ACH receptor on the motor end plate. So here the various receptors are available for the acetylcholine. Now for the acetylcholine we are having the two types of receptor. One is muscarinic receptor and another is a nicotinic receptor. So in the muscles, in the skeletal muscles, we are having the nicotinic receptor, and into the smooth muscles we are having the muscarinic receptors. So in case of the muscarinic receptor we are having five types of receptors, that is M1, M2, M3, M4 and M5. And in case of the nicotinic receptor we are having the two types of receptors, that is NM and ND. Now here you can see the binding of ACS to the receptors causes the new action potential to be generated along with the muscle cell membrane. Immediately after it binds to its receptor, ACH will be broken down by the anticholinesterase. This is the enzyme which is responsible for breakdown of your acetylcholine. So it is an enzyme present in the synaptic cleft. Now here you can see the action potential and electrical impulses travel down the axons of the motor neuron to the end bulb of synaptic terminals. So here in this fashion, so from this axon to the end bulb to the synaptic terminals, the neurotransmitter will be released. They will attach to the muscle fibers, and here the vesicles containing the acetylcholine. This acetylcholine will be released at the synaptic cleft, and at this cleft you can see the receptors are available. So it will bind to the respective receptors. It won't bind blindly, so it will bind to the specific respective receptor of acetylcholine. So in this fashion the further neuroinnervation will occur, means further impulse generation will occur, and it will transmit from one neuron to the another neuron. Now here the action potential causes the synaptic vesicle to fuse with the end bulb. Now it will fuse to this end bulb and resulting in the release of, here it has been release of acetylcholine has been occurred into the synaptic cleft. Now in third position, the acetylcholine diffuses across the synaptic cleft and bind to the ACH receptors on the motor end plate. Now at the motor end plate the various ACH receptors are available to which the acetylcholine will bind to the specific receptor available at this end plate. The binding of acetylcholine to its receptor causes a new action potential to generate along with the muscle cell membrane. Immediately after it binds to its receptor, acetylcholine will be broken down by anticholinesterase. It is an enzyme which is responsible for breakdown of our ACH. Now physiology of skeletal muscle contraction. Now this question always asked in the examination. What is the muscle contraction physiology, or give an account on the process of muscle contraction or how muscle contraction occurs in our skeletal muscle explain and elaborate with the help of diagram as well as with the help of the theory. So here the various points are there. So try to recall all the points. Point wise we have to understand how the process is going on into our muscle. Once the action potential is generated at the motor end plate, it will spread like a electrical current along the sarcolemma of the muscle fiber. The action potential will also spread into the T tubulins, exciting the terminal cisternae of the sarcoplasmic reticulum. This will cause calcium ions get in the sarcoplasmic reticulum to open, allowing calcium ion to diffuse into the sarcoplasmic. Now the voltage gated calcium ions will release the calcium from this sarcoplasmic reticulum, and after this the calcium will bind to the troponin. The whatever the free calcium which has been released from this sarcoplasmic reticulum, now it will bind with this troponin on the thin microfilament and causing it to change its shape. It will change its shapes. Then it pulls tropomyosin away from the active sites. So what is the tropomyosin is there? It will go away from the active sites for myosin. So to this actin, the tropomyosin has been binded already, but when the calcium bind with this troponin, the tropomyosin automatically disclose this actin, and this actin now is ready for binding with this myosin, and it will produce the contraction. So in this fashion this contraction will occur. See, the exposure of these active sites allow myosin to bind with the actin. Means our primary goal or our end point is that myosin should bind with the actin. Then and then the contraction will occur. See here, the contraction has been occur. So the bands has been come closer. There has been overload on one another and causes the sliding of the filaments. The filament has been slided one over the another. Now in details we will see the calcium ions gets in the SR sarcoplasmic reticulum opens and allowing calcium ions to diffuse into the sarcoplasm. So the calcium ion which will release from the sarcoplasmic reticulum, and now they are ready to get into your sarcoplasm. Now they are in their action. The calcium will bind to the troponin. Now this calcium, troponin is here. That free calcium which comes from the sarcoplasmic reticulum, it will bind with this troponin. This is the thin phenomena, thin microfilaments, and causing it to change into its shapes. So it changes its shapes. Then this pulls tropomyosin away from the active sites, from the actin molecules. So here is the tropomyosin, this one. So this tropomyosin is above actin, and due to this binding the tropomyosin will lose its hold over the actin, and after that this actin will be free. This is the active sites, actin. So the actin molecule, it is also protein, so that actin is going to be free, means that tropomyosin will be released. Now here you can see it has been released. Now the myosin heads are energized. Now what are the myosin heads are here? You can see this is the myosin head, this is the myosin head, this is the myosin head. So the myosin heads are energized by the presence of ADP and phosphate at ATP binding site. So it can be energized by this ADP and the phosphates. Once the active sites are exposed, the energized myosin heads, this is the energized myosin heads, so they hook into actin molecules, means immediately they will bind with this actin molecules and forming the cross breeding. So this is called cross breeding. This is called cross breeding. So they will bind with this thin filament of the active site for myosin, which is called actin, and that actin will actively bind with this myosin and they will form the cross bridge. It is called cross bridges. Using the stored energy, the attached myosin heads pivot toward the center of the sarcomere. The ADP and the phosphate groups are released. The ADP and phosphate group will be released from the myosin head. A new molecule of ATP bind to the myosin. Now again you can see the new molecule of ATP will bind with this myosin and causing cross bridge to detach from the actin strands, and the myosin head will get re-energized as the ATP and ATP plus phosphates. So after that these myosin heads again will re-energize and it will release this ADP and phosphate molecule again. As long as the active sites are still exposed, the myosin heads can bridge again to the next active sites. So again if it is exposed, again it is ready. Now you can see here again this site is ready to bind with the next active sites of actin. Again this myosin will bind with the next actin. So in this fashion further contraction of episodes will be proceed, means after a few interval again next contraction will occurs. Here you can see, so again it will bind with the next actin. So in this fashion the constant chain of this contraction and relaxation, means constant chain of this binding of myosin with the actin will occurs. Now we will discuss regarding the physiology of skeletal muscle contraction with the help of the release of acetylcholine, how the acetylcholine is having great role into our process. So by step we can discuss here. Now this is the next step. If there is no longer action potential generates on the motor neuron, now we are moving toward the relaxation process. Now our last step is here relaxation. In early step we have discussed regarding the contraction. Here you can see this is the contraction has been occur. So it occurs into the 1 2 3 4 5 6 steps. Now we will proceed towards the relaxation, how relaxation occurs. Now here, now into the sixth step, if there are no longer action potential generation on the motor neurons, no more ACH, no more acetylcholine will release. Now acetylcholine will stop to release. Now this anticholinesterase will remove ACH from the motor. Now whatever the acetylcholine has been released here or it which has been remain here, so it can be destroyed by this anticholinesterase, and the action potential transmission on the muscle fiber will end, and because only this acetylcholine is responsible for this action potential, and if there is no acetylcholine means there will be no action potential, there is no action potential transmission, there is no further neuroinnervation will occur. So next step is, so this sarcoplasmic reticulum ruptures, so the calcium ion gets in the sarcoplasmic reticulum will close and the calcium ion will be actively transported back to the sarcoplasmic reticulum. So because cytoplasmic reticulum is storehouse of the calcium, where the calcium can be stored back again, so as the calcium gets in the SR will close, so voltage gated that gets of calcium is going to be close, and due to which that it will go back toward the sarcoplasmic reticulum. Now next is active sites converted, no cross bridge of interactions, due to which with calcium ion removed from the sarcoplasm and from the troponin, the tropomyosin will recover the active sites again, because due to this removal of tropomyosin the active sites was disclosed, it was remained open. Now due to removal of calcium from this troponin, again this tropomyosin will bind to the active sites of actin, and due to which next the contraction will ends. So no more cross bridge, because this cross bridge is responsible for the contraction, for the overlapping of the microfilament of thin with the thick, so it doesn't occur, so no interaction will be there, so no cross bridge interaction will be there, due to which no contraction will occur, and that's why last stage is relaxation occurs. So thin filament slide back to their resting position, resting state, and due to which the relaxation occurs into our these motor cells, into the muscular cell. So in this fashion, it is a complete process of contraction and relaxation. The skeletal muscle fibers shorten as thick filament interact with the thin filament and sliding occurs. The trigger for contraction is calcium ions released by the sarcoplasmic reticulum. When the muscle fibers is stimulated by its motor neurons, the contraction is an active process. Relaxation and the return to the resting length is entirely passive. These physiological processes describe that happen at the cellular level, how the skeletal muscle fibers contracts. But what about at the organ level? How do skeletal muscle like your bicep or branchi contract to create useful movements? Now here, the skeletal muscles are made up of thousands of muscle fibers. A single motor neuron may directly control a few fibers within a muscles or hundreds of thousands of muscle fibers. All the muscle fibers contracted controlled by a single motor neuron constitute a motor units. The size of the motor unit determines how fine the control of movements can be. Smaller motor units precise control, for example, eye muscles. The larger motor units gross control, for example, leg muscles. They are moved by side or in any directions. Recruitment is the ability to activate more motor units as more force need to be generated. Hypertrophy, it is a stressing a muscle. When you are doing the exercise, we are stressing the muscle, we are giving stress to the muscles, causes more microfibrils to be produced within the muscle fibers, allow more cross bridge, means more contraction, resulting into more force strength as well as larger size. So when in the gym, when you are doing more and more exercise, so in that case more cross bridge will occur and more contraction and relaxation of our muscle will occur. So there are always some motor units active even when at the rest. This creates a resting tension known as muscle tone, so which helps stabilize bones and joints and prevent atrophy. What is atrophy? Atrophy is nothing but the when the organ size is going to be decrease, when your muscle size is going to be decrease, atrophy. So they are developing our muscle tone, muscle contraction. Anatomy of muscular tissue. Origin, the muscle attachment that remains fixed. Insertion, the muscle attachment that moves. And the action, what joint movement a muscle produces, for example flexion, extension, abduction. For the muscle to create a movement, they may only pull, not push. Muscle in the body rarely work alone and are usually arranged in the functional group surrounding a joint. A muscle that contracts to create desired action is known as the agonist or prime mover. A muscle that helps the agonist is a synergist. When we are helping, when you are moving in one direction, so it will be called as synergistic. The muscle that opposes the action of the agonist, therefore undoing the desired action, is called antagonist. So when you are doing the opposite action, it is called antagonist. Now skeletal muscle movements. So now there are various types of movements are available into our body. Now in that, first is flexion and extension. Flexion is nothing but the when we are going to be decrease the angle of joints. So for example, our any joint is a joint between neck, so that when we are decreasing that angle of that joint, so it is called flexion. And when we are increasing the that angle of degree between that two joints, so that it is nothing but the extension. Now abduction. Abduction is nothing but the when we are going towards the midline of our joint in our body, so it is called abduction. When we are going away from the midline, so it is called adduction. For example, you are raising the hand and you are moving a hand towards our body. Next is rotation. So when you are doing the rotation of our neck from right to left, it is called rotation. And internal, middle or external rotation, when you are going external part laterally. For example, you are moving the hand laterally at the lateral and middle position. So it is called rotations. So our joints can be rotated from one place to the another place. Next is pronation and supination. What is pronation? The pronation is nothing but the when we are moving anything to an anterior position anteriorly. For example, when we are moving our jaw at anterior level, so it is called pronation. And what is supination? It is exactly opposite position of that. So when we are moving back our jaw at the normal position at the back side, so it is called supination. Now elevation and the depression. So when we are going to be elevate our body parts, so it is called elevation. And when you are going to be depress, it is called depression. Next is protraction and retraction. So protraction is nothing but the when we are superficially we are moving our any part of our body, so it is called as protraction. So when we are moving our jaw at superior position into the lateral direction vertically, when you are moving it upward, so it is called protraction. And when you are moving it downwards at the inferior position, it is called retraction. Now next is dorsiflexion and the plantar flexion. So dorsiflexion, when we are moving the our ankle joint, when we are moving that foot at the upper position, so it is called dorsiflexion, means at the above, at the outer position. And when we are moving that ankle, when you are moving that ankle joint, when you are moving that foot at the lower or to the normal position, so at downward, it is called plantar flexion. Next is inversion and eversion. So inversion is nothing but the inside. So when your palm size or your palm or your foot or your sole, when you are moving at the midline at the below position towards midline, inversion inside our leg inside, so it is called inversion. And when we are moving towards the midline to that foot or palm at the outside, so it is called eversion. So these are the various movements when our joints are doing. Now these are the names of skeletal muscles, which you can see various types of muscles are here. It is just impossible to read each and every muscles. Now again, here is the overall of the muscles, how many muscles are there at which part. Adds in the beginning we have discussed. For example, this is the gracilis muscles, abductor longus, tibia muscles. This is the muscles from the back side which are available here. So this is the gluteus muscles through which you are getting the injections. Here the deltoid muscles, sinus muscles. So students, there are lots of muscles are available in our body. Try to recall out of them few muscles. And also here the important topic is the physiology of contraction of our muscles, neuromuscular junctions, how they are working. I hope whatever points were discussed today that you understood and digested well. If you have any issue regarding the muscular system, you can ask me at any time. So please subscribe my channel, like my videos, and share as much as you can. And if you have any queries regarding my video, you can ask me in my comment box. So see you then for today. So we will see next part in the next lecture. Okay, see you then, bye.